Good morning, everyone. My name is faaiq. Now I am going to tell about primitive cells. What are primitive cells? Primitive cells, also known as unit cells. These are the smallest repeating units of a crystal lattice that retain the symmetry and properties of the crystal. Let's discuss what are the characteristics of primitive cells. Smallest repeating unit. Primitive cells are the smallest units that can be repeated to form the crystal lattice. Symmetry. Primitive cells retain the symmetry of the crystal lattice. Lattice parameters. Primitive cells are defined by their lattice parameters such as edge length and angles. Type of primitive cells. Simple cubic. A primitive cell with a cubic shape and one lattice point per cell. Body-centered cubic. A primitive cell with a cubic shape and two lattice points per cell. Face-centered cubic. A primitive cell with a cubic shape and four lattice points per cell. What are the importance of primitive cells? Crystal structure. Primitive cells help describe the crystal structure and symmetry. Material properties. Understanding primitive cells is crucial for understanding material properties such as electrical and thermal conductivity. Material science. Primitive cells are a fundamental concept in material science, helping researchers design and develop new materials. Essential components. Here are some essential components. Depending on the context, essential components can vary. Here are few examples. Central Processing Unit (CPU). It executes instructions and performs calculations. Memory (RAM). Temporarily stores data and applications. Storage drive. It holds operating system, programs, and data. What are the essential components of a research paper? Introduction. Introduces the research question and context. Methodology. Describes the research design and methods. Results. Presents the findings. Discussion. Interprets the results and draws conclusion. What are essential components of a business plan? Executive summary. Provides an overview of the business. Market analysis. Analyzes the target market and competition. Financial projections. Estimates revenue and expenses. Management team. Describes the key team members and their roles. Other contexts where essential components are crucial include engineering. Components like sensors, actuators, and control system. Biology. Components like cells, tissues, and organs. Project management. Components like scope, schedule, and budget. Now we will see about self-assembly and compartmentalization. Self-assembly and compartmentalization are fundamental concepts in biology, chemistry, and material science. Self-assembly. Self-assembly is the process by which individual components spontaneously organize themselves into a more complex structures or pattern without external direction. Characteristics of self-assembly. Spontaneous organization. Components assemble with external direction. Thermodynamic stability. The assembled structure is stable under specific conditions. Reversibility. Self-assembly can be reversible, allowing for disassembly and reassembly. Examples of self-assembly. Biological systems. Protein folding, lipid bilayer formation, and virus assembly. Material science. Self-healing materials and supramolecular polymers. Compartmentalization. It refers to the organization of biological systems into distinct membrane-bound compartments that perform specific functions. Characteristics of compartmentalization. Membrane-bound components. Compartments are separated by membranes. Specific functions. Each compartment performs a specific function. Regulation. Compartmentalization allows for regulation of biological processes. Examples of compartmentalization. Cells. Organelles like mitochondria, nucleus, and endoplasmic reticulum. Organisms. Tissues and organs with specific functions. Synthetic systems. Artificial compartments such as vesicles and microactors. Importance of self-assembly and compartmentalization. Biological systems. Essential for cellular function, regulation, and organization. Material science. Enables design of complex materials and systems. Nanotechnology. Allows for creation of nanostructures and devices. These concepts have far-reaching implications for understanding biological systems, designing new materials, and developing nanotechnology applications. Now we will see about protocell formation of key theories. Protocell formation is a crucial step in understanding the origins of life. Here are some key theories. Theories of protocell formation. Lipid world theory. It suggests that lipids played a central role in the formation of protocells, providing a membrane-like structure. RNA world hypothesis proposes that RNA molecules served as both genetic materials and catalysts, potentially giving rise to protocells. Prebiotic vesicles. suggests that vesicles formed from prebiotic molecules could have served as primitive cell-like structures. Iron-sulfur world theory. It proposes that iron-sulfur clusters played a key role in the emergence of life, potentially contributing to protocell formation. What are key factors in protocell formation? Membrane formation. The formation of membrane-like structure to enclose genetic material and metabolic processes. Genetic material. The emergence of genetic material such as RNA or DNA to store and transmit information. Metabolic processes. The development of metabolic processes to sustain life. Challenges and open questions. Origin of genetic material. How did the genetic material emerge and become encapsulated within protocells? Membrane stability. How did early membranes maintain stability and selectively permit the passage of molecules? Energy sources. How did the protocells acquire the utilized energy? Research directions. Experimental simulations. Laboratory experiments simulating early Earth conditions to study protocell formation. Theoretical modeling. Mathematical modeling of protocell formation and evolution. Synthetic biology. Designing and constructing synthetic protocells to understand the minimal requirements for life. These theories and research directions aim to shed light on mysterious origins of life on Earth. Protocell growth and division. Protocell growth and division are essential processes for understanding the emergence and evolution of cellular life. Protocell growth. Protocell growth involves the increase in size and complexity of protocells potentially through membrane expansion, incorporation of new membrane components, genetic material replication, replication of genetic material, metabolic activity, increase in metabolic processes. Protocell division. Protocell division involves the separation of protocells into daughter protocells, potentially through membrane fission, splitting of the membrane, genetic material segregation, distribution of genetic material to daughter protocells, mechanism and challenges, self-replication. Protocells must replicate their components and divide. Stability and regulation. Protocells must maintain stability and regulate growth and division. Research directions. Experimental models. Developing experimental systems to study protocell growth and division. Theoretical frameworks. Developing theoretical models to understand protocell dynamics. Synthetic biology. Designing synthetic protocells to study growth and division. Implications. Origin of life. Understanding protocell growth and division can provide insight into the emergence of life. Synthetic life. Developing synthetic protocells can lead to new technological applications. Now we will see about metabolism in protocells. Metabolism in protocells refers to the chemical reaction and processes that occur within these primitive cells-like structures, enabling them to sustain like functions. Key aspects of metabolism in protocells. Energy sources. Protocells may utilize various energy sources such as chemical components or light. Nutrient uptake. Protocells may take in nutrients from their environment. Waste removal. Protocells may eliminate waste products. Chemical reaction. Protocells may facilitate chemical reactions such as synthesis or degradation of molecules. Type of metabolism in protocells. Autotrophic metabolism. Protocells produce their own food through chemical reactions. Heterotrophic metabolism. Protocells rely on external source of nutrients. Challenges and open questions. Energy coupling. How do protocells couple energy sources to drive metabolic processes? Metabolic regulation. How do protocells regulate metabolic pathways? Research direction. Experimental models. Developing experimental systems to study metabolism in protocells. Theoretical frameworks. Developing theoretical models to understand metabolic processes in protocells. Synthetic biology. Designing synthetic protocells with function metabolism. Implications. Origins of life. Understanding metabolism in protocells can provide insight into the emergence of life. Synthetic life. Developing synthetic protocells with function metabolism can lead to new technological applications. Encapsulation of genetic material. It refers to the process of enclosing genetic material such as DNA or RNA within a protective structure such as vesicle or a protocell. Importance of encapsulation. Protection. Encapsulation protects genetic material from degradation and damage. Concentration. Encapsulation can concentrate genetic material, facilitating interactions and reactions. Compartmentalization. Encapsulation can create a defined environment for genetic material to function. Methods of encapsulation. Lipid vesicles. Lipid vesicles can encapsulate genetic material. Polymer-based systems. Polymer-based systems can encapsulate genetic material. Protocells. Protocells can encapsulate genetic material. Current research and experiments. Lab synthesis. Creating protocells from basic molecules artificially. Behavioral studies. Observing protocell response to different conditions. Complex function. Incorporating protein synthesis into protocells. Significance and future applications. Origin of life. Provides insight into how life began on Earth. Drug delivery. Protocells can also deliver medicine directly to target cells. Biotechnology enables creation of synthetic life forms for new uses. The future of protocells. Unique origins insight. Protocells clarify life's mysterious beginnings. Medical revolution. Potential to transform treatments and therapies. Ongoing research. Continued study is key to unlocking potential. Thank you.